Good morning, students. In today's class, we'll discuss about plant viruses as vectors. So now, again, I'm going to Unit 1. This is one topic which we have not discussed in Unit 1. In the previous classes, we have discussed several types of these cloning vectors, but these plant viruses we have not discussed. So in today's class, let's see what are the different plant viruses that can be used as cloning vectors, what are the properties, advantages and disadvantages. So there are certain criteria of these plant viruses to use them as cloning vectors. If you look at these plant viruses, one attractive feature of these plant viruses is that not only the intact virus particle can infect the plant, even the nucleic acid which is purified from this plant virus is also infectious. That is one attractive feature of these plant viruses, because of which we can use this plant virus as cloning vector. But it doesn't mean that any plant virus can be used as a cloning vector. As I told you, there are certain criteria that needs to be fulfilled by this plant virus so that we can use it as cloning vector. So what are those criteria for a good vector? That virus should be able to spread from cell to cell via the plasmodesmata. Not only it is infectious, it should be able to spread from one cell to another cell via plasmodesmata. These plasmodesmata are the channels that connect the cells in the plant. Then the viral nucleic acid that must be able to replicate and spread from cell to cell in the absence of viral code. So, you all know, if you look at the virus structure, there is genetic material, whether it is DNA or RNA, and that is encapsulated by viral code protein. But to use this plant vector, to use this plant virus as vector, the viral nucleic acid should be able to replicate and also spread from one cell to another cell in the absence of virus code. That means the nucleic acid alone should be able to spread from one cell to another cell. And this modified viral genome, what do I mean by modified viral genome? We all know that during this recombinant DNA technology, what do? We insert our gene of interest or foreign DNA in the cloning vector, and that recombinant DNA molecule we introduce into the host organism. So the modified viral genome, whichever we introduce into the host organism, that should either elicit very little or no disease symptoms. You all know these plant viruses, they cause different types of diseases. The virus which we are using as a cloning vector, after the modified genome is introduced into the host organism, which means that is plant, it should not cause any disease symptoms in the plant. That is very important. Otherwise, the plant will get the infection and thereby the disease. Sorry to interrupt. I think there is some disturbance. I think somebody has switched on their mic. Let me check who is that. So there is a lot of disturbance because of that. I think Tejashwini. Yes, she muted herself. I think now we are good to go. Okay, so the modified viral genome should either elicit very little or no disease symptoms, and this virus, whatever we select as cloning vector, that should have broad host range. It should be able to infect the very wide range of plants, so that we can use it in number of plants as a cloning vector. And preferably the virus whichever we select, that should have DNA as genetic material. You also know that viruses could contain either DNA or RNA as their genetic material, but preferably we want the virus to contain DNA as its genetic material. The reason why we want DNA containing virus is that the manipulation in vitro at DNA level is easier compared to the RNA molecule. That means the genetic manipulation of DNA is easier. We have very well established methods for DNA manipulation in vitro. So these are all the criteria a virus needs to fulfill so that we can use that virus as a cloning vector. What are the criteria? It should be able to spread from one cell to another cell via plasmodesmata, and the nucleic acid of this virus should be able to replicate and spread from one cell to another cell even in the absence of viral coat, and the modified genome should not elicit any disease symptoms. It should have broad host range, and preferably it should have DNA as their genetic material. So as I told you, as the last criteria, preferably the virus should contain DNA as their genetic material. There are two groups of plant viruses. They contain DNA as their genetic material. One is collimomosaic viruses. Other group is Gemini viruses. So let's see what are the properties of these two groups of viruses and how they can be used as cloning vector. First, we will see the collymomosaic viruses. One important candidate or member of this collymomosaic virus group is CAMV. This is cauliflower mosaic virus, is one of the member of this collymoviruses group. Obviously, they are the DNA containing viruses, and the DNA present is double-stranded circular DNA. And one unattractive feature of this virus to use as cloning vector is it has got restricted host range. That means very limited number of plants are infected by this virus, and there are no non-coding regions in the genome. Why we are interested in non-coding regions? If the non-coding regions are present, those regions would be the ones can be replaced by our gene of interest or foreign GNA. But here in case of cauliflower mosaic virus, we do not see any non-coding region in the genome. There are only two genes, that is gene 2 and gene 7. They seem to be non-essential for infection. So there are no non-coding regions present in the genome. Definitely we have to replace any coding region only. When you see all the genes which are coding the proteins, it seems like gene 2 and gene 7 are non-essential for infection. Obviously, you have to look for some non-essential genes so that the virus replication is not disturbed. So in that scenario, we have to replace either gene 2 or gene 7 because they are non-essential for infection. So one such approach has been demonstrated by replacing the gene 2 with the bacterial methotrexate resistance gene. So this is one demonstrated example using this cauliflower mosaic virus as the cloning vector. The gene 2 was replaced with bacterial methotrexate resistance gene, and the host plant what they have selected was turnip plants. So when such experiment was done on turnip plants, not only these turnip plants became systemically infected in the sense, the virus which infects the plant spreads to all the cells. So not only the turnip plants became systemically infected, they also developed resistance to methotrexate when it is applied in the form of spray. That means this approach was found to be successful. Which approach? The non-essential gene 2 was replaced with a gene of interest. Here the gene of interest considered was bacterial methotrexate resistance gene, and the host plant selected was turnip plant. But one major limitation with this cauliflower mosaic virus is its capacity. If you introduce the longer foreign DNA, it cannot be packaged into virus particle. There is very smaller size limitation if you take cauliflower mosaic virus as a cloning vector. So these are the properties. This is how the cauliflower mosaic virus that belongs to colimoviruses group can be used as cloning vector. Next group of viruses are Gemini viruses. As I told you, these are other group of viruses that contain DNA as their genetic material. Why these are called Gemini viruses? Because of their geminate morphology. That means they occur as paired particles. If you look at the genetic material, it has got single-stranded circular DNA as their genome, but the replication takes place via double-stranded intermediate. We have seen many such examples. Though the genetic material is single-stranded circular DNA, the replication takes place via double-stranded circular DNA as its intermediate. One advantageous or attractive feature of these Gemini viruses to use them as cloning vector is that they've got wide range of crop plants. That means when you use these Gemini viruses as cloning vector, the host plant range is very wide because this virus can infect a broad range of plants. So these are the two groups what we have discussed about the plant viruses containing DNA as their genetic material and their properties. If we look at the viruses, plant viruses that contain RNA as their genetic material, what is the possibility? To what extent we can use this, RNA containing plant viruses as the cloning vectors? The main difficulty or challenge of using these plant viruses containing RNA as cloning vectors is the joining of RNA molecules in vitro. We all know, and I mentioned in the beginning of this class also, the manipulation of genetic material, that means the manipulation of DNA in vitro conditions is easier compared to the manipulation of RNA in vitro conditions. So there is a difficulty of joining RNA molecules in vitro. That's the reason generally these RNA containing viruses are avoided as cloning vectors. But if you are able to make cDNA copies of this RNA prior to the manipulation, uh, we can try using these RNA containing plant viruses as cloning vectors. So one such approach has been tried with the gene that encodes resistance to chloramphenicol. So the chloramphenicol resistance gene had been introduced into Brome mosaic virus genome to check whether the RNA containing viruses can be used as cloning vectors or not. The strategy or method seem to be definitely possible, but using this cDNA intermediate is slightly found to be cumbersome. Though it is possible technically and theoretically, but when you come to the practicality point of view, the method seem to be slightly cumbersome. But if you are able to make it possible, the range of potential vectors will be expanded because most of these plant viruses are RNA containing viruses. If you are able to make this approach uh, possible, then the range of these potential vectors will be expanded. You don't have to restrict yourself only to those two groups of viruses containing DNA as their genetic material. Any virus, plant virus, though it contains RNA as its genetic material, that can be used as a cloning vector if you are able to use this particular strategy. Which strategy? Making the RNA into cDNA and then go for manipulation. Thereby you can use that vector as a cloning vector. And another attractive feature of these RNA containing plant viruses is they have got filamentous morphology. That means they are rod shaped. So in case of filamentous morphology, the size of the virus particle is determined by the length of the viral DNA. That means there will not be any size restrictions on the nucleic acid to be packaged. So you don't have to restrict the size of the foreign DNA that has to be incorporated into the vector, because it has got the filamentous morphology. Whichever the length of the nucleic acid, that determines the size of the virus particle. So without any size limitation, any size of the foreign DNA fragment can be introduced into the viral genome. So these are all the good properties of these RNA containing viruses by which you can use them as cloning vectors if you are able to make cDNA copy out of the RNA before manipulation. What are the disadvantages of using these plant viruses as vectors? So the disadvantages would be, as we all know, the key requirement for plant breeding is whatever the genes we are introducing, that needs to be transmitted to the subsequent generations. This becomes difficult when you use this virus vector if the virus is not seed transmitted. So if the virus is not seed transmitted, in that case, the modified plants, that means the genetically modified plants, they need to be vegetatively propagated. This is all right in case of the plants that can be vegetatively propagated, like your potatoes. If those plants are not vegetatively propagated, in that case, the genetic information whichever is introduced, or the genetic information whichever is carried by the virus, that will be lost if the virus is not seed transmitted. So in that case, it would be more satisfactory or advantageous if the gene of interest could be introduced into a plant chromosome. Whatever the gene of interest that we introduce, if that gets integrated into the plant chromosome, that could be the best cloning vector, especially when it comes to the point of plants. So now my question to you is, what is such vector we studied which can be used in plants and that integrates the gene of interest into the plant genome or plant chromosome? Are you there, students?
Yes, ma'am.
Yeah, tell me, what is that vector we have studied earlier that introduces your gene of interest into the plant chromosome? Do I need to call any name, or will you answer voluntarily?
Ma'am, TMV.
TMV. That's virus, right?
Yes, ma'am.
First of all, TMV is RNA containing virus, isn't it?
Yes, ma'am.
Yeah. Just
TI
now,
plasmid, ma'am.
who is that? Akshay?
Yes, ma'am.
Yes, TI plasmid is a vector we studied in the earlier classes, which is used for the transfer of genes into the plants, and that integrates the gene of interest into the plant genome or plant chromosome, so that that will be passed on to the subsequent generations of the plants. That is why the most preferred vector to use for genetic engineering or genetic manipulation of plants is TI plasmid. Okay. Yeah. So that is all I wanted to discuss in today's class. If you have any questions, please ask me. Otherwise, I will continue in the next class with the gene transfer methods. Okay. If you have no questions, I'll call attendance.